Thanks for having me. Thanks for coming. Yeah, so I gave it a funny title. I thought that might attract some attention, maybe not. So when I think of tissue mechanics, I think about the interaction of mechanics, biology, and material science. And my path to where I am right now started in mechanics and mechanical engineering. My first job was in biomechanics. Then my master's and PhD were more materials science type projects. And then in my postdoc, I got into a lot of biology. And I guess an issue I had was that we talked a lot about translation and getting findings out from bench to bedside type of concept, but I just didn't see it happening. It wasn't applied enough. And then fortunately, one day while I was working in the lab, a gentleman, the manager of the tissue bank at Mount Sinai Hospital where all this work was done, he came and said, we think we have a problem. We've been told we have a problem, but we're not really sure. And so the problem was that that tissue bank and many tissue banks use gamma irradiation to sterilize the tissues that they harvest from donors who have left their bodies to medicine through, in Ontario, it's the Trillium Gift of Life program. And so that donor tissue will be processed and irradiated for sterilization, and then eventually, once selected, will be implanted in a host in the patient. So the thing is they use 25 to 35 kilograms of gamma irradiation to do the sterilization. It's very effective in killing bacteria and some viruses. But they were having product quality concerns, concerns about weak and brittle bone in particular, and complaints and concerns from orthopedic surgeons and other users. And so I went to the literature and I had a look, and even in the last decade, there's been attention paid to this, even though gamma sterilization's been around for a while. And you see that it's out there, especially in the tissue mechanics and biomechanics literature, where we see loss of strength, loss of ductility and toughness in tension, and more importantly, loss of fatigue resistance to even 1% of normal, and loss of fracture toughness in mode 1 tension. And this is all at the conventional tissue banking dose of 25 to 35 kilograms. So I really, we need to assess this more, being a researcher and wanting to know about the impact of this problem. I didn't call it needs assessment at the time, but it was. We wanted to look at, from the clinical perspective, does this matter in orthopedics where we might be concerned with reconstructing the skeleton? And where this really does matter is large skeletal defects. And so these can result from bone cancer, trauma, maybe in revision of joint replacements. And the problem really is critically sized defects that they just don't heal. They're too big for the biology to handle healing. And as an example, this is an X-ray of a patient with a tumor in their femur. The arrow points to, it's not very easy to see, but it's there. And a surgeon, an orthopedic oncologist, would treat this with a radiation, but they'd also eventually cut out a large volume to remove the tumor. And then you have what's called a segmental defect. And the challenge is reconstructing that. And so one of the things they use is large metal, what are called mega prostheses, just large titanium alloy type implants. They are used, but they're mainly used in older patients where they're not very ambulatory, and you're not going to expect a long lifetime for the device. But what about, well, cancer, bone cancer doesn't discriminate by age. We see it in 15-year-olds, everything, all age groups. So the other option is allograft, and so that's the donated human bone. And it is used clinically. That's why we have tissue banks in some hospitals. And there is some potential for revitalization and remodeling in the very long term. So yeah, we're talking about intercalary, in this case, femur reconstruction. So here's an X-ray from a colleague at Mount Sinai Hospital, and he's an orthopedic oncologist. And so you see the femur here. The graft actually is not very visible. It's gray, as is the bone, but it's inside this construct with metal plates and screws and even methyl methacrylate filler, bone cement filler to try and reinforce it. And the failure modes for these kind of reconstructions are not extremely well studied, but the big names have documented non-union, so the bone and the graft are not growing together. Infection, resorption, so the graft actually is just resorbed by the body, by the bone destroying cells called osteoclasts, and then by fracture, and that's a pretty high rate, 20 to 40%. And so what we're really talking about is using a piece of dead bone as an implant. And what comes with that is that you have the need for osteoconduction, the host bone, to grow into the implant, into the dead bone, but this is really slow. And so you're essentially dealing with a dead bone implant, and it is a microcracking material. So over time, under physiological loading, it will accumulate microcracks and eventually fracture. So we can see how, if we use a bone graft that's even more brittle due to sterilization, you have a material selection error here. You will really want a better material to put into these patients so they don't microcrack and eventually fracture. And so here's evidence of that microcracking actually does happen in vivo in human bone. Oops. So yeah, so we're back to this. The problem of irradiation sterilization, and there's one clinical retrieval study that has shown that using irradiation sterilization seems to double the fracture rate over just fresh frozen large cortical bone grafts. So it's a problem for orthopedic surgeons, it's a problem, so that's clinically, and then it's a problem for industry in terms of the tissue processing, tissue banks industry. So how does this irradiation cause this problem? The losses in mechanical properties, and to address that we need to know a bit about bone. And so cortical bone is what I'm focusing on for this talk, and so that's the dense bone and the shafts of the long bones. And it is a composite at the very fine nanoscale of hard, brittle, very stiff hydroxyapatite mineral, and then the soft, tough organic phase, which is mainly type 1 collagen. And so we can see this here. And when we think about composites, we're more concerned about volumes, and so it's about equal volumes of mineral and organic phase with some water. And so the collagen molecules, these rods here, and the mineral particles come together at the nano level. These are about 40 nanometers long. And so this is the type of information we need to know about really where does bone get its toughness from. And if we look at this Ashby plot, we see elastic modulus, called stiffness in this figure, and toughness, so fracture toughness. And we see that we're combining hydroxyapatite, which is very stiff but not very tough, with collagen, this red dot, which is quite tough but not very stiff. And we get this nice mixture in bone where you gain an order of magnitude in stiffness but only lose half an order of magnitude in toughness. So really the mineral is gaining greatly in toughness. So again, how does irradiation cause this problem? Well, we and another group in the United States have looked at this, and basically we did biochemical and some biophysical techniques looking at protein fragmentation, solubility, what happens to the thermal mechanical behavior of the collagen, and really what we ended up with in our paper is confirming the obvious loss of toughness and strength, but also understanding that it's through actual fragmentation of the collagen network by the gamma radiation, actually fracturing the collagen molecules, that leads to the loss of connectivity and all these other results. So you can read about that in this article further if you wish. So if we were to develop a needs statement to solve this problem, we might have said that we develop a means of protecting the connectivity of the bone collagen network during irradiation sterilization in order to protect the mechanical properties and improve clinical outcomes. And ideally not impacting upon the effectiveness of the sterilization process, the host graft union rate, and osteoconduction. But of course, at the time, we weren't actually that sophisticated. We were asking a more simple question of what if we could just maintain the connectivity of the bone collagen? Would that help? And so we had a hypothesis that pre-treating the cross-linking with a cross-linking agent before radiation would improve the connectivity. And this was, and therefore the mechanical properties. And actually this was a bit counterintuitive based on some information in the literature, because if you take normal bone and you cross-link it, it actually becomes brittle. But I'd been, at the time, reading a lot about the effects of aging and disease on collagen in bone and what it does to the mechanical properties. And it turns out with age that ultimate strength and elastic modulus don't actually change that much. What actually changes a lot with age is the amount of strain that the bone can accommodate before fracturing. So this is severe loss in ductility in a very strong relationship. And it turns out that in Peter Ziopoulos from England, quite a while ago now, he measured connectivity using a thermomechanical technique, which I'll get into later. And he showed that the connectivity of the collagen decreases with age. And so that's a lot like what happens with irradiation sterilization. And more importantly, for this talk, what he showed was there's a positive correlation between connectivity and work to fracture. And I want you to remember that idea because it's pretty important to the rest of this talk. And so what you ended up seeing was younger donors up here and older donors down here. So, but a strong, and an even stronger correlation with fracture toughness. So that was a key idea. So we weren't just sort of hypothesizing out of nowhere. We had an idea that if this holds true for, in the case of irradiation, we could use this idea, try and protect the connectivity of the bone collagen and achieve the results we were seeking. And at the same time, I was studying the effect of over cross-linking on bone using ribose. And it turns out this is actually a really good means of basically over cross-linking the bone before irradiation. So we have tried other agents, but ribose is the one that works. And I'm not going to go into all that work, but it's small. It actually will diffuse into the dense matrix of bone. It's relatively fast cross-linking kinetics. The products are pretty biocompatible. One of the cross-links that ribose forms when it reacts with collagen is called pentosidine, and it's found in vivo, although it's found in situations of aging and disease. So we're still working on whether this is the best option. And it's not known to protect against the effects of gamma radiation in the sense that a free radical scavenger might. And so we think that's a good idea because we don't want to actually protect the bugs and the viruses. We just want to protect the bone. And so early on, about 3 years ago now, we showed that it worked using ribose, pre-treating the bone with ribose and then irradiating. It worked quite well. We used radio-labeled ribose to measure how much ribose was actually being incorporated into the bone matrix, and then measured 100% protection of ultimate strength and 60% protection of work to fracture and ductility. And these are some representative curves here from 3-point bending. Oh, and you can read about that here. So the real question now, we're getting, at this point we were getting some interest from the intellectual property people at the hospital, and could this actually be turned into something commercializable? And so the real question is, will this actually work in human bone? Because cows and humans are not the same, to put it very basically. But so we looked at pre-treating human cortical bone with ribose in phosphate buffer solution at our most ideal conditions from a lot of trials beforehand. And the idea being that it would also protect strength, ductility, toughness, and that there would be this relationship between work to fracture and connectivity. So the benefit of working in a hospital and one with a tissue bank is you can ask them to be collaborators, or it was kind of a mutual thing, but because they wanted a solution to this problem or would like to know if it was possible. And so they donated us human femurs, and we used males to control for things like osteoporosis and stuff like that. And then from the shaft, we were able to cut out portions located anatomically, and then in each basically plate of bone that we're cutting out of the cortex, we would produce adjacent beams called a match set, and within each match set we'd have a control, which was just fresh frozen after being cut, a conventionally irradiated control, and then a number of treatment groups. And so this is sort of the experimental design: cortical bones treated with ribose, irradiated, and then we did our mechanical testing and collagen analysis for connectivity. And we had multiple ribose groups, plus or minus irradiation, and our controls. So we use match sets to enable us to control for interlocation and inter-donor variability, because that's quite significant. And we just we did 3-point bending after the specimens returned from gamma radiation, and we follow an ASTM standard that other people in the literature follow. 3-point bending is not ideal, but it gives us a starting point for the mechanical testing, and we measured things like elastic modulus, strength, ductility in terms of strain to failure, and work to fracture. So how much work did it take to break the beam divided by its cross-sectional area. And after that, we took the 2 halves of the beam, and 1 half was decalcified gently decalcified in sodium or EDTA solution, and that just gently chelates out the mineral phase, and you're left with a strip of decalcified bone collagen. And then we use a thermomechanical technique called hydrothermal isometric tension testing to get a measure of thermal stability and connectivity of that network. So it's clamped between 2 clamps, and the water bath around it is heated up at a controlled rate from ambient to 90 degrees. And the device, this is a drawing of the device we've built. So we have our specimen with it between grips, some fixtures, and then a load cell, and it's temperature controlled. And so as we heat from ambient, which would be around 25 degrees, water coming out of the tap basically, it will eventually hit a temperature where the collagen is driven to melt, and that temperature will shift depending on degradation of the collagen structure. But more importantly, it will, once it's driven to melt, it was also driven to shrink. But because it's held at a fixed length, it'll actually generate a force that we can measure and convert to a stress. And we can use this slope, or the maximum slope in this range, as a measure of the connectivity of the collagen network. So we tried a number of different ribose concentrations in this experiment, from 0.06 all the way up to 1.2. The ribose solution is getting pretty syrupy at this concentration. And so you see here, these are stress strain curves from the 3-point bending. And so we have our control in blue, our irradiated control. You see the loss in strength and toughness, and then a gradual increase in protection, especially at 1.2 molar. And for comparison, there's 1.2 molar without the irradiation, just to show that by itself, the ribose protection treatment doesn't seem to do very much. So really promising results: 100% protection of strength, like we saw in the bovine bone, and protection of work to fracture. So that was very exciting. And when we evaluated the stability and connectivity of the bone collagen, we have stress versus temperature. Here's blue again is the control. And you see the drastic effect due to irradiation. It starts to melt at a lower temperature. It generates a much lower slope, so that's a lower measure of connectivity, and it actually will rip out of the grips because it's weaker. Whereas when we apply the increasing concentrations of ribose, we're able to bring the slope back up. The thermal stability is pretty similar. And so we've effectively protected the collagen network. That's not to say that it is normal collagen at all, but the connectivity and the stability have been protected. And if we plot normalized toughness versus normalized collagen connectivity, we see this relationship, which is actually very reminiscent of that aging work that I told you about earlier. So we think we have a good idea about how this system works. So now for the stuff I've not presented before. I just had a master's student who finished in September, and he was tasked with measuring fracture toughness rather than just strength and ductility, because of course fracture is what we're really concerned about. This is how these grafts actually fail, due to the dead brittle bone accumulating microcracks, then eventually becoming a fracture. So I'm going to just go over basic fracture mechanics really quickly. Cracks act as extreme stress concentrations, so as you move closer to the tip of a crack, the stress will, in a linear elastic fracture mechanics, approach a singularity, infinite stress, which of course the material won't do. And in reality, the material will not sustain a stress, say, in a perfectly plastic, elastic, perfectly plastic material. It won't sustain a stress higher than, say, the yield stress. And so what happens is you get a region of material in front of the crack that is yielding, and it's called the process zone. And in bone, that's made up of lots of microcracking and plastic deformation, and it results in a redistribution of the stress. And it's large enough in bone that the process zone is large enough in bone that you can't use linear elastic fracture mechanics. And so what we end up using is, for these reasons of the process zone forming microcracking ahead of a crack tip, this is all happening at the very small scale, down sub-nano scale, microcracking of the mineralized collagen, plasticity in those fibrils, and microcracking. We end up using elastic plastic fracture mechanics to try and capture some of the irreversible effects. And it's an integral. It's called the J integral. It goes around. You integrate around the crack tip. Fortunately, experimentally, it reduces down to a very simple expression when we're using what are called single-edge notch bending specimens. And this can actually be divided out into an elastic component and an irreversible component. And so J is our fracture toughness measure, and it's the nonlinear version of G, which is from linear elastic fracture mechanics, which many of us are familiar with if we come out of engineering, some branches of engineering. So bone gains its fracture toughness from mechanisms that happen ahead of the crack tip and also mechanisms behind the crack tip. So what we call intrinsic ahead of the crack tip and extrinsic behind the crack tip. And they act at different scales. And so I already mentioned microcracking and plastic deformation ahead of the crack tip. And then what we also see is mechanisms like collagen fiber bridging behind the crack tip. And these apply tractions. They basically apply stresses that they steal energy and driving force away from a crack tip under load. And then also very significant is crack deflection. So as the crack changes direction, the driving force to grow that crack changes. And so bone has all these mechanisms at play. And collagen, changes in collagen will affect both, we think, intrinsic mechanisms and extrinsic mechanisms. So the result of the extrinsic mechanisms is that you'll get what's called a rising R curve. So fracture toughness in a material like bone isn't a single point measurement. you don't just load it up and it catastrophically breaks. There will be a stage where you'll reach what's called J1C, crack growth initiation fracture toughness. And then the mechanisms within the bone will actually allow the crack to grow in a stable manner before you get catastrophic failure. And so we wanted to capture this rising R-curve behavior because it's relevant to how well bone or bone graft material can accommodate cracking and resist catastrophic failure. And so what do you need to generate R-curves? You need your measure of J. You need your force versus load line displacement curves. And you need some measure of the crack extension as you're testing. So our hypothesis for all this would be that we'd have very idealized JR curves as such. Normal bone being tougher over a range of crack lengths, irradiated bone being negatively affected by the irradiation, and then the ribose treated and irradiated would be somewhere in between. Excuse me. And so, similar to the last study, but with far less groups because of the dimensions of our specimens, we took the diaphyseal cortical bone, machined out sets, in this case 10 sets, from, again, male human donors, and the same type of design, but doing fracture testing instead. And so we use the single edge notch bending configuration. So it's basically 3-point bending again, but with a notch machined into these specimens. We do this with a diamond wire saw, which is 300 microns in diameter. We end up with a semicircular end here, and then we sharpen it with a razor blade to get a very fine notch with the radius in here at the tip is around 5 microns, so it's very fine. And then we cut the specimen so that the crack is going to grow transverse across the bone direction, or across the long axis, which is most relevant to fracture, and then also in the circumferential direction. And that's mainly because if you try to grow the cracks in the radial direction, they deflect drastically, and it's very hard to do the testing. So that's, and then we also, because we needed to track the crack growth, and we decided to do that optically. We used a black, actually permanent marker, just to create some contrast, as you'll see. Actually, could you turn the lights down in the front a little bit, please, Christina? Thank you. So the test setup, we use an Instron benchtop, it's called an Electropulse E1000. And so we have our 3-point bending configuration in here with the load cell. And then all this contraption over here is a macroscope for 32 times magnification and a 25 megapixel digital camera in order to capture crack growth as the test is running. So we had a timing chip, which was triggered by the mechanical testing machine, and also the pulses were captured, so we knew the timing of the pictures, LED lighting, and then the digital macroscope capturing the specimen. And so from our JR curves, we needed some metrics, and today I'm just going to talk about J1C mostly. So that's the crack initiation, detectable crack initiation, crack growth initiation fracture toughness. So an ASTM standard that's used quite widely in this field is called E1820, and it tells us to use an offset line. And this is mainly just to make sure it's detectable by actually other techniques that we're not using, because bone doesn't behave like metal, basically. And so we define this metric as our crack growth initiation fracture toughness according to ASTM. And then we also did it optically by trying to capture the instant when we could see crack growth. And that was actually quite challenging, because here's no crack growth, and here's crack growth. And it's this tiny little white line here. So it was very difficult. High resolution, but still very difficult to see. So here are some example tests that you can see the specimens are moving. They're deflecting under the load that's applied through this nose. And you'll see that the irradiated specimens, the crack starts growing much earlier than the other two groups, and it grows much faster. And in fact, this specimen will fracture completely while these two are still, the crack is still growing in a stable manner. Yeah, see that test is done and these two are still going. The normal bone is doing better than the ribose-treated bone, but much better than the irradiated group. And so this was just accomplished with our mechanical testing machine and capturing pictures with our macroscope. And this gives us the information to generate our own JR curves. And here are some representative curves. So J versus crack growth. These are the force versus load line displacement curves if you're interested in those. But as we expected, so we have the normal bone in the solid line. The dotted line is irradiated, and then the circles and dashed line, that's the ribose pretreated and irradiated. So you can see the protective effect over a range of crack growth. As I mentioned, I'm going to talk mainly about J1C. Turns out that at very small crack growth, using our optical method, we really couldn't statistically draw out differences in the means. But when we use the ASTM method, which allows for a little bit more crack growth, we get this separation. And in fact, we get 75% protection. It's about 50% loss here from normal to irradiated, and then 75 protection. And statistically, these two are not, we couldn't separate them, but we have the statistical difference here and here with very small p-values. Then just to confirm the effects in the collagen again, these are the HIT curves. Normal and ribose-treated and irradiated are almost the same. And there's your irradiated group failing early with 80% protection of the melting temperature and 93% protection of the connectivity measurement. So in summary, ribose pretreatment partially protected the transverse fracture toughness and rising R-curve behavior. This seems to be through an effect more on the extrinsic toughening mechanisms than the intrinsic, because we didn't actually see that detectable difference in the observed J1C rather than the ASTM. So we're gonna work on that a little bit more because we think it's important to bone fracture mechanics in general. And Robert Ritchie's group from Berkeley, they argue quite often that bone relies more on its extrinsic mechanisms anyway. So that seems consistent with their findings. And our findings are consistent with our previous work and the idea of protecting the collagen connectivity. So all that has resulted in, we filed a patent, well, the hospital did, over a year ago now. And we're now arguing with the patent office about it. And so we're the first to show that this could be done, both in bovine and human bone. And actually, it's the idea of pre-treating bone with ribose to accomplish this is actually counterintuitive or counter the literature, because there are studies out there who've used ribose to make bone brittle as as a model of disease. So we get plenty of questions about it. And then the idea is this will lead to improved clinical outcomes and reduce healthcare costs as we try to avoid large reconstruction failures due to fracture. So right now, I have a master's student back in Toronto who is finishing up our first study in vivo, where we used a rabbit model to actually do a segmental defect and reconstruct it with normal irradiated and ribose and irradiated. It's going really well. Better than expected results, and he's been invited for a podium presentation in March at the Orthopedic Research Society. We're very excited about that. And we did some work with bacteria to see what effect the ribose pretreatment has on effectiveness, and it seems to have no effect. It seems to have killed all the bacteria, which is great. And there's more mechanical testing to do. I have a PhD student in Toronto who is working on that, and I'd like to do some actual fatigue crack propagation rather than just quasi-static fracture. And if we learned anything in doing this thesis with my master's student was that we actually do need some better methods, because taking standards from fracture testing of metals and applying it to bone just doesn't make a lot of sense, and there are some considerations that need to be, well, I'd like to see standards, but we'll see if that ever happens. And then I'm getting into some finite element modeling with a new colleague here, John Montesanto in mechanical. And the adventures in commercialization will continue. And for new directions, coming here, trying to start something new, the question is, can we solve the large defect problem by design and actually build and design and build bone biomimetic materials to better meet the need, rather than trying to just return to the normal mechanical bone properties, because normal bone, if it's dead, just doesn't make a good implant anyway. So we've got improvements, but I believe things could be better. And that is my talk. Thanks for your attention. As you can imagine, there's lots of people involved in this kind of work, especially the tissue bank and my mentor, Mark Grimpus, while I was at Sinai. And I must acknowledge funding from CIHR, especially from the Institute for Musculoskeletal Health and Arthritis. They gave me my first grant and have continued to give me money as a young investigator. So I really appreciate that, of course. So thank you, and questions, the good part.
How do you explain, for example, to the reviewers that your results somehow in direct it's contradictory to what you have saying in the literature that the material is used to actually make bone brittle?
Yeah, so the ribose embrittlement model's been around for, I don't know, 20 years, let's say, maybe 25. The key difference is when you apply ribose to, when others have applied ribose, they've done it under different conditions. We use high temperature, we use 60 degrees. So maybe the reaction products are different. Maybe we're forming more crosslinks versus adducts, that kind of thing. And also they're applying cross-linking to bone that has normal levels of connectivity, whereas we're applying it and then we're damaging the connectivity. So I think by radiation.
So radiation actually, this is an interplay.
Yeah, exactly. So in a sense, we're compensating for loss of connectivity due to radiation. We're over cross-linking ahead of time to compensate for that. And so that's the explanation. In the other models, the connectivity is just elevated above normal. And so that suppresses the ability for the bone, collagen, to actually deform plastically and absorb energy. It becomes itself linear elastic and more brittle. Yeah. So actually this fracture stuff is in review right now in the Journal of Mechanical Behavior of Biomedical Materials. And I'm looking forward to a good review on that. Yeah.
Thank you. It's been reported that due to radiotherapy, although it's much lower radiation than what you reported here, some incidents regarding the ribs fracture.
Mm-hmm.
So it's much lower than what's been reported.
Yeah.
As far as the energy concerned.
3 orders of magnitude lower.
That's right.
Yeah.
And so do you think that the site of the bone affect the sensitivity of the bone itself to the radiotherapy, to radiation, sorry? And also, how about the age of the bone, how much mature it is?
Yeah, great question. 2 great questions. The first is we actually see those fractures induced by irradiation therapy in the femur as well and in other bones. So it's not necessarily site dependent, but it's probably through different mechanisms, because with that dose of gamma, you're not going to induce as much collagen fracture or scission. I've actually worked on that problem with Peter Ferguson and Mark Grimpus in vivo, doing some in the rat. And it seems to be the damage to the cells, actually. It's the lack of repair. So, of course, that isn't completely proven either, but that's the preconceived conclusion, which I shouldn't have, but that's how I see it working, is that it actually it's killing the ability to repair the microdamage, then you get accumulation and fracture by a similar mechanism. So the question about the maturity of the bone is really good, actually, really, really good, because I left that part of the story out of my presentation, and it's in the paper. Actually, what we see in bovine bone is the effect of gamma, the same dose, the effect is much larger in bovine bone than in human bone. And one of the potential reasons is that bovine bone is a lot younger and has a lot higher connectivity measurements. So in a sense, there's farther for it to fall when you irradiate it. And so we see 70% loss in fracture toughness rather than 50, just out of the air numbers, but that kind of proportion. And therefore, it's actually easier to compensate for the damage as well. So we get less protection in bovine and more protection in human. Now, all the human bone we've used has been from older patients, so 55 to 60. We actually haven't tried, say, a 20-year-old or 30-year-old, because that bone actually gets turned into grafts. It's because it's healthy and really good graft material. So I suspect we'd see the same thing, but we haven't done that study. Yeah. The thing is with aging, you get a lot of damage in the collagen itself just due to aging and disease and chemical processes and remodeling. Yeah. Any other questions? Well, yeah, ask them while we have coffee. All right. Yeah, thanks a lot.